Assalamu alaikum wa rahmatullahi wa barakatuh. Hi everybody. Today inshallah we'll speak about the histology of the respiratory system, and we will start by the upper respiratory tract. As you know, we will divide the respiratory system into two parts. The first part is upper respiratory tract. Second one will be the lower respiratory tract. So the learning objectives to overview the components of the respiratory tract, describe the function of the conducting portion of the respiratory system, and then we will compare the characteristic microscopic feature and function of the different region and airway of respiratory system, including the lining epithelium, the gland, cartilage, smooth muscle, and elastic fiber. And we'll speak about any correlated clinical conditions with the knowledge of the histology of respiratory system. So first of all, we need to know the component of a respiratory system. So the respiratory system has two portions: conducting portion, which is responsible to conduct the air from and to alveoli, and also responsible for the warming and the humidify and filter of the air going inside the lung. And the conducting portions including the nasal cavity, the pharynx, larynx, trachea, bronchi, and bronchiole. And also we can see also including as extra pulmonary, which nasal cavity, pharynx, larynx, trachea, primary bronchi, and some of the intrapulmonary is also still as conducting portion. So secondary bronchi, bronchiole, terminal bronchioles, they are intrapulmonary structure. However, this still considered as part of the conducting portion. The second part is the respiratory portion, and this is responsible for gas exchange, and this portion include respiratory bronchioles, alveolar ducts, alveolar sacs, and the alveoli. And of course, all the structure will be intrapulmonary structure. So if you look at this diagram, you'll find this is the conducting portion for nasal passages, and then larynx, trachea, right? And you can see here the trachea, and then primary bronchi, secondary bronchi, and secondary bronchi is still it became intrapulmonary. However, it is still conducting portion. So also bronchiole, terminal bronchioles, until this one, yes, it is intrapulmonary, but still conducting portion only to conduct the air. And the gas exchange start from the respiratory bronchioles, alveolar ducts, alveolar sacs, and alveoli. So you can see now the difference between the conducting portion as yellow parts, and also the respiratory portion is the blue parts. So now if you look at the human diagram, so you will find now what the air, and when you inspire air, there's something should be happen to the air before enter the lung. So you need to slow the airflow, also warming the airflow, moisten the airflow, and also filter and remove the particles and sense odor in the air. And also we have part for phonation, which is speaking. And once it reaches the lung, what will happen? We'll have gas exchange, removal of the particulate matters, and also for acid-base balance. So now let us see what should happen and what structure which help for that action. So when the inspired air coming inside, before it reach the lung, it need to be slow. So a slow of the airflow usually happens by the presence of the turbinate bone. As you know, the nasal cavity has turbinate or concha, right? It would be differ according to species, and this is conchi or conchi will help for slowing of the airflow before going to the lung. Also, you need to warming the airflow. And nasal cavity consists of the venous plexus, which is responsible to warming the airflow before going to the lung. Also, you need to moisten the air, and this will happen by the serous gland, which is present in the nasal cavity, to moisten this air. Also, we need to filter the blood, we need to filter the air before going to the lung. And who will do that? We have mucus gland or goblet cell, which is important for drop particles, and also we have the cilia, which lining the trachea is important, right, to remove the the foreign particles to the pharynx, and also we have alveolar macrophage present in the lung. All this will help to filter and remove particulate matter. Right. For a smell, as you know, olfactory receptor present in the nasal cavity for the olfaction. Another function needs for the air movement, which is phonation, which happens by the vibrating of the vocal folds. And also you need to have support of the conducting structure, so you need bone and cartilage support, and also the trachea will play a role in the ability to vary flow of the air through the tracheal muscle, which is present in the trachea. So now let us take general information about what is the structure of the conducting system. So the conducting system, right, or conducting parts, right, which including nasopharynx, including the neurons, nasal cavity, right, until the terminal bronchioles, they are conducting system. So we need to speak about the epithelium, the respiratory epithelium lining this structure, and also the lamina propria, the mucus and serous gland, cartilage, smooth muscle, and adventitia. So as you can see now from this diagram, you can see it is covered by pseudostratified columnar epithelium, which has goblet cell and it have cilia, and you will know now the importance of this structure. So now if you take what is the cells which are present in the respiratory epithelium. So there are many cell types present in the respiratory epithelium, including the ciliated columnar cells right here. So ciliated columnar cell is abundant and is important. This cilia they are important to move the mucus and also for trap the particles into the oropharynx. In this case, you have to make expectorant of to make expectoration of this one to be expectorated outside the body or to be swallowed inside the digestive system. So now the cilia is very important for trapping and for to move mucus and trapping the particles. Also, the epithelium has goblet cells. Goblet cells is important to to secrete mucus, is important for moistening and also for trapping of the particles. Also, the epithelium has basal cell. Basal cell here is a basal cell right here. This cell is important for replacement of the cilia and also replacement of the epithelium, including the goblet cells. Also, you have brush cells, and the columnar brush cells here is no cilia, but it has microvilli, and this is usually connected by the nerve ending, right? Sensory receptor associated with the trigeminal nerve, which is number 5. Also, the epithelium includes a small granule cell. This is granule cell important to regulate the airway and diameter of the blood vessels, and also for the mucus secretion. So now again another photo for the conducting airway. So you can see now we have the pseudostratified ciliate epithelium here, and also we have goblet cells in the cell. And then you can see here you have a basement membrane right under the epithelium, and then you have lamina propria, and lamina propria, as you can see it in this photo, right under the basement membrane, and it connective tissue and blood vessels. Then under the lamina propria, you can see we have here the smooth muscle. We have the smooth muscle here, right? And then you have the submucosa, and submucosa has important gland, ceromucous gland right here, right? And you can see also we have the cartilage. So the importance of the gland, as we mentioned, to moisten the air and also to prevent the dryness of the alveoli. Right. Another photo for the epithelium. Again, you can see the pseudostratified epithelium here, right? Pseudostratified, right? You can see now the pseudostratified ciliated epithelium, and then you have goblet cells right here, and also you can see the basement membrane, and also a lamina propria containing, blood vessels here. So let us start by the part also again. Let us continue by the parts of the, the conducting portion. So we have the nasal cavity. And as you know, nasal cavity has respiratory epithelium and also has olfactory epithelium. So respiratory epithelium is covered the entire part of, of the majority of the respiratory, of the of nasal cavity. However, the olfactory epithelium will cover the ethmoidal turbinate bone. Also, nasal cavity contains serous and mucous gland and also blood vessels. And as you know, we have a nasal septum. So if you look at this one as the turbinate, you can see here the nasal cavity, cross-section nasal cavity. You can see here we have the turbinate bone or concha. We have a dorsal turbinate and you have the ventral turbinate, and then we have the olfactory region. We have the ethmoidal turbinates will be in this area as olfactory region. So if you look at also here transverse section nasal cavity, you can see also we have the dorsal nasal turbinate and we have ventral nasal turbinate, and also we have the nasal metasis. We mentioned the importance of the turbinate bone to make interruption, and this is why to make slowing, right? To interrupt and slow the laminar air flow. What purpose of that? The slowing of the air is important for warming and moistening of the air before reach to the lungs. Again, if you look at this one also in the nasal concha or nasal turbinate, is typical feature of the conducting portion. Then we have pseudostratified ciliated epithelium here, and then we have pseudostratified columnar ciliated epithelium has goblet cells, and you can see also lamina propria submucosa, and we could contain also mucoserous gland. Again, in the nasal concha turbinate, again you can see the pseudostratified columnar ciliated epithelium. We have goblet cells here, and also we have the mucoserous gland, typical structure of the conducting portion. As we mentioned before, the glands are important in humidifying the inspired air and also to prevent the dryness of the alveoli. Also, another photo to show that is, the nasal concha has venous plexus, and we mentioned in the beginning the venous plexus is important for warming of the inspired air. So let us now speak about the ethmoidal turbinate, which is important. Ethmoidal turbinate, which is covered by olfactory epithelium and usually passing through the cribriform plate right here. So as you know, olfactory epithelium is especially present in well-developed in certain animals such as dogs. So you can see also the olfactory epithelium here is pseudostratified columnar ciliated epithelium, and also we have a lamina propria, and it's important that it have nerve fiber to conduct here, for olfactory nerve fiber, and this one is important, as olfactory epithelium is important because dogs can use for for discovering illegal drugs, as important to for finding missing person, right? And also for many security. So dogs has well developed olfactory epithelium, right? So one of the important structure here that the olfactory epithelium here has different types of the cells. So you can see now we have the basal cell, you have olfactory cell, and you have subarticular cell. Usually this is subarticular cell is important to support, right? Is important for for supporting, right? And also you can see here we have basal cell, and the basal cell usually, right, for replacement of the olfactory cells and subarticular cells. Also, you can see the olfactory epithelium is covered by non-motile cilia, because this is important because for smelling, and also it has olfactory serous gland. This gland is very important because it moistens the surface epithelium, right? And also, when the animal smells the odor, this one will be furnished, right, an aqueous liquid fluid to dissolve the substance, to allow, after dissolving, to allow the substance to binding with, right, with odorant binding protein, which will be carried now to receptor, right, to recognize the smelling. And also the secretion of the olfactory gland is contain also lysosome and IgA. Right. Also, one of the important structure here is the vomeronasal organs, which present, well developed in certain animals such as dogs. So if you look at this diagram, you can see here, this is vomeronasal organ here is covered by two types of membrane. Respiratory membrane would be, or respiratory epithelium would be in the lateral aspect, and we have the olfactory epithelium in the other aspect. And as you know from the anatomy, we have the incisive duct. So the vomeronasal organ, it is present, at the base of the nasal septum, and usually connected with, this one connected with, by the incisive duct. So incisive duct connected between oral cavity and nasal cavity. Oral cavity at the incisive papilla, and in nasal cavity usually joining with the vomeronasal organ. Just you have to know, in case of animals such as horse, usually the oral end of this duct, the incisive duct, will be like blind. The importance of the vomeronasal organ is for sexual behavior and kin recognition, right? So it's very important also for dogs. Also this and this why it is well developed organ. Right. Paranasal sinuses, from anatomy, it is present in certain bones of the skull, such as frontal bones and also such as maxillary bones. And paranasal sinus usually, as you know, it's connected with the nasal cavity, and the lining by simple squamous or copoidal epithelium until became pseudo-stratified columnar epithelium as respiratory epithelium. And this one has ducts which has glands or goblet cells, right? And usually the lining continue with nasal cavity, because there's some connection between paranasal sinus and the nasal cavity. Pharynx now, as you know, we have oropharynx and nasopharynx, and usually we have dorsal to the soft palate, and this one will be covered by pseudo-stratified ciliated columnar epithelium. It became by going caudally, it became as stratified squamous epithelium. Also, as pharynx containing tonsils and also lymph nodes. So you can see now nasal cavity, oral cavity, we have nasopharynx, we have oropharynx, we have the laryngopharynx here, the larynx, trachea, and this will be the esophagus. Right. So from gross anatomy, we know there's many cartilage present in the larynx. So we have thyroid, cricoid, epiglottis, arytenoid. So some of these cartilage will be elastic cartilage, some will be hyaline cartilage. So hyaline cartilage such as thyroid and the cricoid cartilage, elastic cartilage such as the epiglottis and arytenoid cartilage. As if you look at this one, you can see now the oropharynx, and you have the larynx here, we have the vocal fold, we have the laryngopharynx, esophagus, and the trachea here, and you can see the epiglottis here, thyroid, and the arytenoid and the cricoid cartilage. So what is the epithelium usually lining or cover the epiglottis? Epiglottis and the vocal fold usually covered by non-keratinized stratified squamous epithelium. And also the only one covered by respiratory would be the false vocal fold. So if you look at this diagram here, and this is one, in the epiglottis, you can see now the epithelium non-keratinized stratified squamous epithelium, and also we have lamina propria submucosa contain the mucus gland. Of course, when you go, epithelium gradually, because when you go caudally now, it will change into the respiratory epithelium, which is pseudo-stratified columnar ciliate epithelium, and also contain, mucus and serous gland, and also contain lymph nodules. So now you can see the different types of the of the cartilage. So the cartilage here is hyaline cartilage here, the epiglottic cartilage right here, the adipose tissue and the elastic fiber, and also epiglottis here. You can see also cross section to see the elastic fiber present in the epiglottis. And one of the interesting structure that is, the epiglottis of the dog usually has adipose tissue, which is unique in canine. Right, now we should do the trachea. Trachea now it is a flexible tube. It is started from the larynx until primary bronchi. As you know from biology, trachea have C-shaped cartilage and lining by pseudostratified ciliated columnar epithelium, and it has submucosa and adventitia, and also has the submucosa contain gland, right? And usually, it also has a tracheal muscle, which is different from species. Some species like, dog present outside the tracheal ring. Some other animal will be the trachea, it will be inside the tracheal rings. So now from this diagram, you can see now, right, we have the trachea has C-shape, right? C-shape hyaline cartilage. And why is hyaline cartilage? We know later on, because to prevent the collapse of the, of the trachea during inspiration. And also to have the gland, right, in submucosa, right, and it has also adventitia right here. And, we have tracheal muscle, which differ according to species. So if you look at, this, here, you'll find here the typical pseudostratified columnar ciliate epithelium has the goblet cell, and lamina propria has, has blood vessels, and we have a mucus gland is important right here. So as we mentioned, why the trachea has C-shape? To prevent the collapse of the trachea and only allow variability in the diameter. Also, you can see now it has goblet cell and mucus gland, and this is important to trapping particles, right? As you know, and also has elastic fiber. Why trachea has elastic fiber? To be flexible, for flexibility. Why the tracheal muscle has trachea? Why the trachea has trachealis muscle or trachea? Why is not a smooth muscle? Is not tunica muscularis is a tracheal muscle, a smooth muscle. Why it has? Because this one is important to narrow the lumen of the trachea, especially during cough reflex, to increase the falls and the velocity of the expired air, right? To narrow the lumen of the trachea. So just now, try to identify the structure, what is the purpose of this type of the epithelium in the system, and also if you can identify the structure. Thank you so much. Inshallah, we'll speak in next part about the the lower respiratory tract. Take care. Assalamu alaikum wa rahmatullahi wa barakatuh.